Thank you everyone for joining us for today's webinar in the CTMC 2016 series. The topic today is going to be small sample size clinical trials presented by Dr. Chris Coffey. I just wanted to start with some housekeeping items before we get started. You'll notice everyone except for the speaker will be muted today, and this is so that we prevent any feedback during the presentation. We would like the presentation to be interactive though, so please use the request to interact live button at the top right of your screen in order to be given the microphone. There's a delay of about 5 to 10 seconds since this is a live feed, so please just wait a couple seconds before you start to speak. If you're interested in CMEs, you must complete an evaluation form, and we'll provide all the full details at the end of the webinar. Right now, I'm very pleased to introduce our speaker. Dr. Coffey is a professor in the Department of Biostatistics and director of the Clinical Trials Statistical and Data Management Center at the University of Iowa. He served as the PI of the Statistical Center for the Secondary Prevention of Small Subcortical Stroke Study, or SPS3, and he's an active member of the NSDK study section. He serves on a number of data and safety monitoring boards and chairs the Education Committee of the Society for Clinical Trials. Dr. Coffey also serves on the executive committee of the Clinical Trials Methodology course. So we're very grateful to have you on, Dr. Coffey.
All right, thanks, Sam. So as Sam mentioned, what I'm going to talk about today is a very high-level overview of small sample size clinical trials, which is a topic that could get really in depth. Next slide, please. So what I want to address here is mainly to focus, at least early in the webinar, on the importance of adequate study planning for these types of small clinical trials, which probably addresses a situation that many students in this course will find themselves in as you flesh out your ideas into a full protocol. And then talk about kind of two broad approaches for dealing with some of these issues: analytical approaches that are important to keep in mind, and then also kind of alternative design options that can be used with small sample size clinical trials. Next slide, please. So just general disclosures, I have no commercial financial interest and don't include any information on unlabeled use of any commercial products or investigational use not yet approved. Next slide. Now for the fun stuff. Next slide as well, but I think Sam already hit on that. So what you see here is a general picture. This is an excuse to get the hills of East Tennessee, which is where I'm originally from, into the presentation. But also, if you'll click ahead slightly, Jenna, what you can see here is kind of an illustration of what in statistics I might refer to as the wonderful land of asymptopia, where as far as you look, everything looks like a bell-shaped curve or a normal distribution. And in statistical terms, and particularly in planning clinical trials, that becomes extremely important because a lot of statistical methods development is based on large sample theory, and things really simplify substantially when you can assume normality. Unfortunately, when you're dealing with small clinical trials, the normality assumptions or these large sample approximations that are often used for these methods fall apart, which makes the planning and the conduct of these trials much more complex. Next slide. I think you'll have to go through several. Perfect. So one of the questions that comes up as I've discussed this topic in the past is exactly what we mean by a small clinical trial, particularly small sample size clinical trial. And I think it's important to note that that often depends on the context and can mean different things to different people. So many might view a small clinical trial as kind of an early phase trial that's needed in order to move a development of a new drug or intervention forward into what would eventually be a larger confirmatory phase 3. So for instance, a stroke researcher might need an early small clinical trial in order to find, for instance, a maximum tolerated dose. their compound that they're going to take forward into proof of concept studies and eventual efficacy studies. While alternatively, a researcher in a rare disease area like ALS or other rare diseases might think of a small clinical trial as what they would need in their confirmatory phase 3 clinical trial. So I'm going to try to address both of these as we move forward. Most likely, you may be in the earlier situation, or you could be in the latter. You could be doing a phase 1 or phase 2 type early study in a rare disease in which you would really have to address a lot of the issues that will come up in the webinar today. Next slide, please. So in general, we have to keep in mind that there's really no magic wand. There's really no simple fix. We want to get the right answer. And it's important to keep in mind that small studies are not mini versions of large studies. And often we have to make some very difficult choices because of limitations in small study. And I'll further illustrate that in a few minutes, but we have to know what we're sacrificing. And it's important to have those conversations up front. And one of the things I think that is somewhat of a catch-22 that many of you may find as you begin to work on your protocols, particularly for those of you participating in the course because you're generally early in your career, is for these types of early trials, particularly when you're challenged by resources like sample size, it's really critical to have connections to the right individuals. A collaborator who might have the clinical trials experience, a statistician who can address the design issues, but that's very challenging for those of you who are early in your career. So one of the objectives of the course is to try to help to develop an infrastructure as you work through your small groups in order to provide that level of expertise to help you address many of the topics that may come up in the discussion today. Next slide, please. So one of the things I want to kind of pause for a moment and look at a document that came out now, it's about, I think, 10 or 15 years old, but there was a committee by the National Academy of Sciences document that prepared an executive summary on small clinical trials, issues and challenges. I think it's important to kind of look at some of the key recommendations from that. Next slide. The first recommendation that came from that was to define the research question. That's really the same as you would do with any clinical trial, and in particular, what many of you are going to focus on in your early calls with your small working groups that you have within the course. And that's really nailing down what exactly is the research question of interest. So how are you going to define the outcome? What are you going to test? How are you going to take the clinical question and put it into something that is testable from a statistical perspective? So that has to be kind of defined, and everything else will kind of fall from that. Next slide, please. The second recommendation that they had was to tailor the design. And so there, it's important that you choose a study design that is set up in such a way that you can answer the question. And so that's going to almost be step 2 of what you're going to go through with your small working groups. It's particularly important in small clinical trials that you really address the design as thoroughly as possible, because efficiency is critical because of the limitations that you may be under. And you want to make sure that you can address the clinical question of interest. One of the challenges that you will come into as you lay out your question of interest and then start to institute a design is how can you set the design up so that you have adequate power to answer the questions. One of the things that is particularly troublesome is if you get to the end of a study and you find out that you did not reject your null hypothesis, but it turns out that you are underpowered to reject it if it were actually true. So if you saw some difference in the intervention you're looking at, is then you spend a lot of time and resources running a study that really never provided a clear answer. Because one important thing that you should always keep in mind as you flesh through the design is that failure to reject a null hypothesis does not imply that the hypothesis is true, because you could fail to reject the null hypothesis either because the null is true or because the size of difference that might exist was not one that your study was large enough to detect. And we often don't know how to tease that out. And it's particularly challenging in these small trials because we don't have large sample sizes that give us the ability to detect small or moderate types of effect size in general. Next slide, please. The third recommendation is really when you go to report the results of the clinical trial, which is way down the road, I recognize for many of you, but it's important to clarify the method. So you want to, if you've come up with a design that's appropriate to answer the question, you want to be clear in how you report that when you report the results either in presentations or publications down the road. Next slide, please. The fourth recommendation is to perform corroborative statistical analysis. This is particularly true when you have issues like missing data. you might have a plan in place to look at missing data, but you probably want to have a sensitivity analysis as well, where you would look at several different alternative strategies for handling the missing data. One of the things you would like to see is that if your overall conclusions tend to be the same regardless of how you address the missing data, that generally strengthens the final conclusions from your study. If you find that your conclusions vary substantially depending upon how you address the missing data, that provides a level of uncertainty and probably suggests it's something that needs to be looked at further. So it's important to have a statistical plan in place that would address all of these issues. Next slide, please. The fifth is to exercise caution in interpretation, right? So anytime you're looking at small clinical trials, I think this is more critical for the space where you're looking at phase 1, phase 2 studies, which is, again, where most of you are going to be looking at in your projects. There's often caution in interpretation, and mainly that is because we tend to go into this with some general sense of what we expect or hope to happen. For instance, if we're looking at an intervention, if you're leading it in a study, most likely you have some reason to think that the intervention is effective, or else you wouldn't be spending your time and resources to do that. What that often means in these studies with limitation is when you start to look at trends, we can often look at, different individuals can look at the same data and see different things depending upon kind of what they want to see. If you're convinced that what you're looking at works, you could look at a piece of data and see a promising trend, while someone who maybe is convinced the intervention does not work could look at the same piece of data and use that to justify the fact that the treatment does not work. So it's important to be cautious in interpreting the results, particularly from phase 1, phase 2 trials. There are numerous examples, particularly in the neurosciences, where there were promising looking results from phase 1, phase 2 trials that end up failing in phase 3. So it's important to be encouraged when you get preliminary results, but it's also important to kind of use a bit of caution and think about the different consequences and how that could be replicated as you move forward in the research design of future studies. Next slide, please. And then finally, the last recommendation was more research on alternative designs is needed. And this kind of gets back to the first 2 points, in that if there's a question you want to address and you want to look at choosing the best way to address that question, when we have limitations in small trials, we might have to consider more innovative or alternative designs, or come up with maybe a standard design, kind of off-the-shelf design doesn't fit as well. There are specific nuances that can be implemented in the design to make it more efficient. And I'll return to that a bit later in the discussion this morning. Next slide, please. So if you look at those 6 summaries, right, these are the summaries that the committee came forward with for their recommendations on small clinical trials. Define the research question, tailor the design, clarify the methods when reporting the trial results, perform corroborative statistical analyses, exercise caution in interpretation, more research needed on alternative designs. Next slide. But when you look at that, that's really no different than what you would see in any other trials. Even in a large sample trial, you're going to have to address those same issues. So that's a fundamental fact that you have to keep in the back of your mind, that even though there are more challenges for small sample clinical trials, the requirements and the things that you have to address are no different than what you would have in other situations. What that means is we're going to have to work extra hard in order to make sure that we can address all of these issues. Next slide, please. So for instance, if you think of, there are 3 basic requirements for any clinical trial, right? It needs to examine at least one important research question that should be clearly defined, as I had mentioned before. The trial should use rigorous methodology in order to answer that question of interest. It's into the plan for the design, how you're going to do the hypothesis test. And then the trial has to be based on ethical considerations and set up in a way to assure that risk to subjects are minimized. Again, these are requirements that would be true for any clinical trial. Next slide, please. What we find, though, is that when we're trying to do a small sample size clinical trial, it becomes extremely problematic to address all of those. And so we may have to relax some of the assumptions. So the design may not be as efficient as we would like it to be, or we can't quite answer the question we want to, we have to modify it in some way. But there's a really big difference between we start to do it thinking we can do everything and we realize that we can't, and we have to kind of backfit things or modify it and figure out what can we do once we've started, versus what would be more ideal is that up front, we understand that there are limitations and we carefully look at the various options that we can use to address those limitations and try to come up with a study design that, if we have to relax some of these criteria, that we have done so in a way that still gives us desirable properties that we wish to move forward, and that we understand what we're sacrificing and what we're gaining, and that we're willing to live with that as a study team. And again, that is what a lot of your discussions in your small working groups may entail. Next slide, please. So there are two general approaches that you can address that, and I'm going to talk briefly about both of these during the discussion today. The first is taking into account the challenges of small sample size studies when you're doing the methodology. And so this gets into the analysis and being as efficient as possible in terms of your methodological approach. Alternatively, the second approach would be to use some type of alternate innovative design. So to try to tailor the design more to the nuances of what you're looking at. Next slide. So first, let's talk about the analytical approaches. So one of the ways to do that is to try to be as efficient as possible in measuring your outcomes and measuring your outcomes precisely. So for instance, one of the things that's well known, if any of you have had any type of statistics course in the past, or you've had any type of experience computing like a required sample size, going through sample size calculations, you know that the detectable effect for a study is related to the ratio of the variance to the sample size, right? So as the sample size gets bigger for a fixed variance, we're able to detect smaller and smaller effects. So if we have a specific effect we want to detect, and we have a fixed variance, in order to get the power that we want, we can just increase the sample size, right? And so if we're studying a population in a moderate or large clinical trial, we can probably find the sample size that's needed in order to address whatever effect of interest is needed. And we can kind of modify that and not have to worry about it too much. There's still a lot of planning, but it's something that is achievable. Next slide, please. If we're in a small sample situation, maybe we can't increase the sample size, right? Maybe we're in a situation where it's just not feasible to do above a certain number because they're either not available, or there are limitations, or we're restricted to a single site, or maybe a small number of sites. So if we can't increase the sample size, the only way to make modifications that would allow us to detect smaller effects would be to decrease the variance of our outcome measures. So we might have to pay special attention to how we're measuring the outcome and how we would capture the variance. That gets into a number of issues. Next slide. For instance, one of the things that could come up is what type of outcome measure are we looking at? There's often different ways to look at outcomes, and there tends to be a bit of a trade-off between what is most efficient statistically and what tends to make more sense clinically. And sometimes there may need to be discussions in making trades between that. For instance, clinicians, in my experience, generally like to think in terms of binary outcomes. For instance, have you achieved a successful outcome or not? Yes or no, defined on some scale. While statistically, we can gain more power with continuous outcomes. One of the challenges with continuous outcomes is you can show a difference in a clinical outcome, or sorry, a continuous outcome that's statistically significant, but it may not be clinically significant. That's one of the advantages of the binary outcomes. If there's a range that can be defined as a successful outcome, it has interpretation if you show it's statistically significant, both from a statistical perspective and from a clinical perspective. But the binary outcome might have less power for a similar situation, and so you'd need to account for that when you're setting it up. That could cause problems, particularly in the small sample size trial settings that we're discussing today. Next slide, please. So as an example, let's say you're going to do an early phase study in ALS. And so the ALS functional rating scale revised might be what you're going to use as your primary outcome. There are different ways that you could set that up. So it's the same outcome, but you're going to look at it in different ways. You could look at it as a continuous measure. So look at the change in the ALS-FRS-R score over some time period. So maybe a change in slope for a treated group versus a placebo group. Or you could treat it as a binary data. So maybe you look at over the treatment period, did the subject have a 10% decrease in their ALS-FRS-R score from baseline or not? So that would define each subject's criteria as a success or not. Or you could define it as a time to event endpoint, where you would look at the time it took each subject to achieve a 10% decrease in ALS-FRS. They'd be censored, obviously, if they did not over the course of the study. So each of these takes the same basic endpoint, but looks at it in different ways. And because it's a different way of looking at it, each of these are going to have different required sample sizes for a specified effect of interest. So one of the things that may need to have some consideration early on is not just what is your endpoint, but how should you measure your endpoint, and how should you capture the endpoint in a way that would go into your test? Should you capture it as a binary data? Should you capture it as a continuous variable? Should you capture it as a time to event variable? How would you do that? That's something that should be taken into account, because that can give you some flexibility with respect to how you can potentially reduce the variance of the outcome that you're measuring. Next slide, please. Another example would be the use of parametric versus non-parametric approaches. So a parametric approach just means that you're using some distributional results. So for instance, if you're using a z-test, you're typically making a parametric assumption of normality. Non-parametric approaches are generally more robust because they don't make distributional assumptions. But what we find is parametric approaches generally have higher power. And so remember, because of the limitations in small trials, a parametric approach would be preferable from a power standpoint as long as the distributional assumptions are satisfied. So this gets back to the point I made earlier where you really need to have a team that's cohesive and understands the area that you're studying and can really have some educated discussions and come to a consensus on how likely they feel that the distribution assumptions are satisfied. So for instance, if you're looking at an area where your parametric approach may require a lower sample size than the non-parametric approach, but if the study team looks at it and the parametric approach, for instance, depends on an assumption of normality, and it's pretty clear in talking with the experts in the field that the data don't look normal at all, then you're falsely gaining additional power because it's not really true because that distributional assumption is not met, and you could end up with bias or other problems from using those parametric approaches. So again, it's a discussion that would need to take place to determine which one best fits the study situation that is under discussion in any specific study. Next slide, please. Another thing to keep in mind is how can you increase power? One of the challenges, so in a usual RCT, right, I mean, there's a lot of complexity in study planning, but from a statistical perspective, you don't have to worry about noise too much. So it's like the runner here who's running with this group of kids behind him. If one of these kids falls down, somebody might notice, but everybody's watching all the kids run. They're not paying that much attention. It's kind of like a standard clinical trial. You want to be reasonably efficient, but a small degree of sloppiness or lack of efficiency is not going to damage the study. Next slide, please. When you're in a small trial setting, it's almost the exact opposite. You really have to carefully evaluate all of your assumptions and be as model-free as possible and pay attention to every piece of data that goes in and try to squeeze every last bit of efficiency out of it. There, it's more like what you see on this slide where you're running by yourself, right? So if you trip and fall, everybody's going to see it and it's going to have great impact, much less than where you had the group of kids all running together. That's more the situation that you have in the small trial setting. Next slide, please. So one of the things that often comes up for discussion is whether you should use historical controls. That's a very hot button issue. Historical controls can be extremely useful when you're comparing a new treatment for these early stage studies. Maybe you can get data from published studies as long as you feel that it's reliable. Historical controls can generally lead to smaller studies because you don't need a concurrent placebo control group, but they have problems. Next slide, please. To use historical controls, you have to be extremely confident that the historical population you're basing your historical controls are representative of who you would get in the study had you put a placebo group in. Things change over time. Approaches to treatment change over time, and there are numerous examples in the literature or from case study experience that individuals have had where studies have used historical controls, but then subsequently found out that those historical controls are not representative of the placebo subjects that would have been enrolled in the studies. This has come up in a big discussion that you may hear in the news in the field of neuroscience today, where the FDA is having the hearing on Monday for the advisory panel to look at the Duchenne drug that's been in the news that's coming up. And one of the aspects of that study, if you read some of the things that have come out about it, is that the comparison of the treated group in there is being addressed to a group of historical controls. And there is some question, depending on who you ask, in that of how representative those historical control subjects are to what would be a placebo. In order, do those historical control subjects look different from the general group of Duchenne's patients that you would see now? So that will be an important part of the discussion that will take place on Monday as that panel reviews that information. But those are the type of issues that you come into. And so in general, in your stage of planning your trials, using historical controls is something you should consider. But I will caution you that there is generally a high bar that it would take to justify using historical controls in place of a placebo group when you're writing a grant application. And so you're going to have to really do a good job from a grantsmanship standpoint in order to rationalize that. Next slide, please. So I'm going to shift a little bit here now to move into some design type issues that you can consider to address this issue. I mentioned you could do this from a methodological standpoint, or you could try to account for this in the design. Next slide. The first one I'm going to talk about kind of morphs both of these together, and that's if you're doing some type of repeated measure design. So for instance, you're enrolling subjects and then you're measuring them over time. So let's say that you're doing a 12-week study. Individuals come in at baseline, they're randomized to drug or placebo. You're going to measure them. They're going to come in every 4 weeks. So week 4, week 8, week 12, you get a measurement. And your primary endpoint is the change in your outcome that you're observing from baseline to week 12. What we would often do in your standard clinical trial is then take the baseline measurement, the week 12 measurement, compare the difference, or compute the difference for an individual subject, average the differences across the groups, and then compare across the groups to see whether we have a difference in the change over time for our treated versus our placebo group. What we don't do, and this gets into some of the, I don't know if sloppiness is the right word, but some of the areas that we don't need to be as concerned about in your traditional trial setting, is we don't really have to perform any type of longitudinal analysis there in order to get that assumption. In a small trial, it might be critical to take into account the longitudinal nature of the measurements. Why? Because by accounting for the week 4 and the week 8 measurements, when we're looking at the change at week 12, we can reduce the variance of our outcome measure. And remember, that was critical because maybe we can't increase our sample size. So the only way to make sure we can detect smaller effects is to reduce the variability of what we're measuring. And so in a repeated measures design in a small clinical trial, you absolutely should consider ways to model your outcome longitudinally and to take advantage of all the outcome measurements that you're collecting on that subject, because you can account for the fact that taking multiple measurements over time in an individual, there is some correlation among those measurements. And so you can learn about future measurements based on the ones that you've made before. And having that as part of your plan can potentially help you squeeze out that extra bit of power that might be needed to make the study viable or feasible. Next slide, please. So for instance, here, this gets to what you're looking at over time. You might consider the final value minus the baseline value. That's a standard approach. The better approach would be the final value with the baseline value as a covariate, or next slide. Or you could look at it where you actually model it over time, right? And that's what I would like to encourage you to move towards. Next slide, please. An alternative design that often comes up for discussion, particularly if you're in a phase 2 setting, would be a crossover design. Generally, in past experiences with the course, there's always been a few students who want to consider a crossover design. That can be extremely beneficial. Again, this gets around the issue of needing a separate placebo group. In a crossover design, each subject is exposed to all treatment. So for instance, if you're looking at a drug versus placebo, all you're doing, you're randomizing subjects, but every subject will get drug and every subject will get placebo. You're just randomizing the order. So you'll have some subjects will get drug first, followed by placebo second, while another group of subjects will get placebo first, followed by drug second. By doing this, each individual becomes their own control. So again, you can reduce the variability through the design because you can compare an individual to themselves. And you're able to look at comparisons with a smaller group because you've got a concurrent control group where each individual is kind of calibrating their outcome in themselves. Next slide. So that is the efficiency that you can gain from this. The disadvantages and the most tricky piece is that you have to be able to measure the outcome, and you can't have a carryover effect. And that's the part that really often limits the use of the crossover design. So what that means is it has to be something that you're treating, that as soon as you withdraw treatment, the individual would go back to kind of their baseline state. So if you, let's say that you have treatment and the individual gets better, if they worsen after treatment, but they don't get as bad as they were before they ever got treatment, then you're going to have differences in your groups based on the ordering. So the group who got drug first, when they get placebo, they're starting from a different threshold than the group that got placebo at the very beginning because they have some carryover effect of the drug that the group got in the first stage. That causes problems with the crossover design because there's no easy way to test for the carryover effect, and you would then need more complicated techniques if you think you have a carryover effect, which often makes the efficiency gain from the crossover design, it limits it because it makes it so much more complex to analyze. So the crossover design is useful, but only if you can make a strong case that the time in between the first stage and the second stage is sufficient, that any effect of treatment in the initial stage is washed out, that it's not carrying over into the second stage. Again, that's something that can be done, but that's usually a high bar for someone in your situation that would have to be justified in the grant application. So if you're considering a crossover design, it would be extremely important to start early on discussions with your your team leaders, your clinical team leaders and your statistical team leaders about any concerns with the carryover effect, and talk very carefully about whether that can be justified or not. Next slide, please. Another situation which is kind of similar to the crossover design, particularly in an extremely rare disease situation, would be an N of 1 design. So an N of 1 design is in essence a special case of a crossover or repeated measures design, where you have a single subject who might undergo treatment over a successive number of periods, and within each period, they get treatment for one part of the pair and the alternative treatment, say placebo, for the other part of the pair. And so you have an individual, maybe, so instead of treating 10 individuals in a crossover design and randomizing the order they get, you have an individual who you're going to randomize the order they get 10 different times, right? So for instance, let's say it's an individual who is prone to asthma attacks, and you might have different ways of treating an asthma attack. You would randomize so that for one pair of asthma attacks, they might get drug first and then some standard treatment second. Then they would get standard drug. You would go for a period of 10 times. Then you combine the data across the different pairs of periods for that subject to get some estimate of, does it look like the subject had a better response to their asthma in the periods of time for which they had treatment versus the period of time for which they had the standard or the placebo. Next slide, please. So you can combine those. If you have a series of N of 1 trials, they can be combined using meta-analysis. So this is something that is typically done for extremely rare diseases. It's something to keep in mind. I doubt that's the situation that many of you are going to be in, but you've seen we've seen N of 1 design start to creep up in the literature and being used more and more. Next slide, please. There are also selection designs. These are kind of slightly different in that there's really no hypothesis test in the sense that we normally think of, but they can be used as kind of a gatekeeping type of procedure. So this would particularly be useful in a rare disease or disease in which there is no approved therapy, but there might be multiple potential therapies that are currently being considered. There are limited resources, so maybe you have limitations from a sample size, and it's just not feasible to separately run a study for each of these. The selection design could be an early phase aspect where you can, in essence, run them against each other in some small study to see which one looks to have the best response. So here, a selection design is kind of like a horse race where you're just putting different treatments together and seeing which one looks the most promising that you could then take and put into a trial and test, for instance, against placebo. So there's really no test here. The type 1 error here, if you're just comparing treatments, is high because you're going to pick one. So you've just decided it's not really looking at efficacy of a treatment. It's trying to get a sense of which treatment looks best, which would be most optimal to take forward into a test for placebo. So it helps with the pipeline problem when there's multiple things to consider. It can be very useful because you can do these in an adaptive nature. I'll talk about adaptive designs in a minute, but you can have a selection design as a first stage that then maybe expands into a slightly larger study that would compare the selected treatment versus placebo to try to see whether there is a sense of efficacy. That has been done in some situations. It's probably a greater complexity than many of you might want to consider for your studies, but it is something that can be out there. Next slide. Another type of design, and you'll hear more about this in the course, is a futility design. So futility design is, you could think of also as a non-superiority design, and it's a screening tool that can help to identify whether agents should be candidates for phase 3 trials. So one of the things that pops up from time to time and really needs to be avoided when you're doing a phase 2 type study is that your question in your phase 2 is the same efficacy question you would see at phase 3. It's just a smaller underpowered version of the phase 3 trial because it's meant to justify getting some parameter estimates that you could use to plan the phase 3 trials. Those tend to get absolutely demolished when they go to study section, and it's pretty clear that an underpowered phase 3 trial is not kind of acceptable from a grant review perspective. A futility design tries to address that in a way that still gives you the data needed to plan the phase 3, but in a way that it can address an important question for the phase 2 study. And so how it is set up is rather than trying to show that the drug is effective, you set up the null hypothesis in a way that assumes it's effective, and you would reject the null hypothesis if the trial is futile, if you've shown it's far enough away from what you said would be the minimum threshold of efficacy that would be needed in order to conclude the drug is efficacious. So if futility is declared, it would imply that the results from the study suggest you should not go forward to a future phase 3 trial. It is extremely unlikely that you would achieve whatever minimum level of efficacy you stated would be important. If futility is not declared, then the trial is not futile. That suggests it should go forward, but you haven't shown that the drug is efficacious. It could be that the drug works, or it could be that there's no difference, or the difference between them was not sufficient to declare futility. You're only going to declare futility if it's much worse from what you've said was your effective interest. Next slide, please. So to put that in context, let's say that you're looking at a treatment where a 10% increase in favorable response rates over placebo is clinically meaningful. So if we were doing a phase 3 trial, our null hypothesis would be that the response rates for placebo and treatment are the same. Our alternative would be that they're different, and we would power that study for the sample size to detect a 10% increase in favorable response rates. A futility design sets it up slightly different. So in a futility design, the null hypothesis is that treatment improves outcome by at least 10% compared to placebo. And the alternative hypothesis is that treatment does not improve outcome by at least 10% considered to placebo. So the farther we are from the 10% increase, the more likely we are to declare futility. So let's say in the extreme example, if the drug actually worsens success rates by 10% compared to placebo, we're going to have a high probability of declaring the study futile. If the treatment actually works by 10% or more, we're going to have very low probability of declaring the treatment futile. And let's say the treatment works equivalent to placebo, we're going to have some reasonable chance of declaring it futile, but we may not necessarily declare it futile. That's what we're willing to live with and would be sorted out in a future phase 3 clinical trial. So that's the futility design, and this is something that you may want to consider in the studies that you're working on in your working groups. Next slide, please. One of the challenges, and this just lays out what I mentioned before on slide 39 of the differences in the null and alternative hypothesis. Next slide. What you may want to keep in mind with the futility design, and I mentioned this, but it's worth reiterating, it has a high negative predictive value. So if you declare futility, then you can be reasonably confident that the treatment is not effective, and that would not justify moving forward in a phase 3 type of trial. But the futility design tends to have low positive predictive values in the sense that not declaring futility does not imply that the treatment is effective. So that's important to keep in mind. So it can really be used when you're in a limited sample size situation, and maybe you're not quite ready to go to phase 3. You need to get some preliminary information to go to phase 3, but you don't have any clear biomarker to look at, so you really need to look at your efficacy endpoint. It gives you a way to address a valid question without simply running an underpowered efficacy trial. So it is something that can be extremely useful. And as I mentioned, there'll be more discussion of this as one of the presentations at the in-person course. Next slide, please. The last thing that I'll mention is adaptive designs. And so adaptive designs kind of incorporate a lot of different components of innovation in that they address the fact that this need for more efficient clinical trial design. And so there's an increasing interest in adaptive designs, which the idea of an adaptive design is you allow reviewing, accumulating information as the trial is ongoing in order to possibly modify trial characteristics, right? So if you find out that some of the assumptions or some of the characteristics of the study, or you start with multiple doses and you drop doses, or you start with multiple drugs and you drop drugs, there's a lot of different ways that you could adapt during the trial, but it's a way to try to be as efficient as possible as we're setting up our trial designs. Next slide, please. A couple of definitions that have recently come out clarify an important aspect of adaptive designs, though. For instance, there's an adaptive design scientific working group that was formed in the mid-2000s. They published a white paper in 2006, which was the first time that a formal definition of adaptive design appeared in the literature, where they defined an adaptive design by clinical study design that used accumulating data to modify aspects of the study as it continues. But then they went on to clarify that it should be done without undermining the validity and integrity of the trial. The changes are made by design, not on an ad hoc basis, and it's not a remedy for inadequate planning. And then FDA in 2010 had a draft guidance for adaptive design in drugs and biologics. A similar guidance came out last year for the devices group. Next slide. But all of these really hinge on this same broad topic, that this whole notion of adaptive by design. So what they have all clarified in the definition is that an adaptive design should be one in which upfront during the design, it is recognized there are some uncertainties, and there's a plan in place in terms of what steps would be taken to address those uncertainty as the data come in. So for instance, if A happens, I will do this. If B happens, I'll do that. If C happens, I'll do that. You have a plan in place. Why is it important to have that plan in place up front? Because that's the only way that you can absolutely show that you preserve the integrity and validity of the study, which again was an important requirement. Because if you specified in advance how you're going to react, you can set up a series of simulation studies under a wide range of conditions, what you expect to happen, what you think is likely but may not happen, and even things that might be the worst case situation that you think is unlikely to happen but could happen. You could address the simulation, you could run through the adaptation and all those properties and make sure that you're getting proper parameter estimates, you don't have any bias in terms of inflated type 1 error, or if you do, make sure that you have a sufficient plan in place to correct for that bias. So again, adaptive designs are something you may hear in the course. It's something that you could consider, but it's going to be a little more complex and is probably not the kind of thing you would want to tackle if you're doing your very first study. In the advanced track, and there's a component of the studies in the course that will exactly get into this adaptive design aspect of it. But for many of you in the foundations aspect of the course, it's important to recognize this. It might be something that you will be interested in or might use down the road. But you could consider it for the trials you're planning now, but I would caution you in that because it might be slightly more complex at this point in your career development. Next slide, please. So to kind of summarize, this was a lot of material, small sample size trials. Again, you could have a whole day workshop on this topic. But the thing I want to particularly leave you with is that regardless of what type of study you're doing, what sample size you have, what disease area you're looking at, you really need an appropriate study design. That appropriate study design should be set up to answer a clear question. It should have a sufficient sample size to answer that question. It should have adequate power as a result of that sample size, and you should have proper control of bias in order to allow a meaningful interpretation of the results. And even though small clinical trials pose important limitations and can induce challenges, we can't ignore these same issues, and we have to use extreme caution to make sure that we're addressing those. And if we have to make any sacrifices in order to address all of those, or maybe we can't completely meet all of them, we need to, as a team, collectively understand what sacrifices we're making and what we're giving up in order to achieve the study that we want to do. As a result of that, additional research into innovative designs for small clinical trials is needed. That's one of the advantages that you have of the course, is that you have at your access, both during the calls and the sessions before and during the in-person course, a series of experts who have done clinical trials who can help you consider maybe some innovative approaches that might be more challenging outside of the developing your early clinical trial outside of the infrastructure of the course. So that's something I think you should definitely take advantage of as you move through the course. And at that, I will pause and address any questions that come up. I see one question: will the PowerPoint presentation be available? And yes, all these will be posted on the course website.
So if there are any questions, please raise your hand. It should be on the right-hand side of your screen, request to interact live, and we'll be able to give you the microphone. You can also use the Q&A box. Okay. I don't see any raised hands so far, and no QA entries so far. So I'd like to say thank you to Dr. Coffey for a great talk, and thank you to all of you for attending today's webinar. Please make sure to complete an evaluation, and especially if you want CMEs, you have to complete a form, and I'll put up the web link in just a moment, but it's bit.ly/R25eval, EVAL. Oh, it looks like there's a couple.
It looks like there's a couple of questions.
Yeah.
Let me start. So there's one about how does futility design address the issue with sample size we were to exclude any null hypothesis, we should be adequately powered. So the futility design works with sample size that's similar from the others in that you choose a different, your null in that case might be no difference or a difference in the wrong direction that you would power the study for, and you would choose your sample size accordingly based on that. So it's functionally very similar. You just have to account for the differences in the way you set up your null and alternative hypotheses. Another question had a question regarding how carryover effect has been addressed in studies looking at behavioral outcome studies. I don't know specifics on that, but I do know that in my experience, it would be kind of challenging to do a carryover effect because with a behavioral outcome study, if you're changing behaviors, there tends to be some residual effects of that change in behavior. So that would be the type of situation where a carryover effect might make a crossover design not completely valid there. For instance, if you're if you're taking a behavioral outcome for, I don't know, just take diet or exercise, right? You're hoping that by doing the intervention, probably someone, so you exercise, they exercise 5 days a week, some specific regimen. After you stop it, I mean, some people will completely stop exercise go back to what they did before, but there might be people who don't exercise as much as they did before the intervention was done, but they might exercise a little bit more, and so they're not quite the same as they were before the behavioral outcome was started. The behavioral intervention was started, and so that could be potentially problematic, and you would have a carryover effect that you would have to account for. There's another question. Can you go into a bit more detail of how longitudinal measurements and standard final minus baseline value differ? Sure. So take the situation I mentioned where you had maybe baseline, a 4-week, an 8-week, and a 12-week measurement. The standard final minus baseline would just take your, let's say you're doing a weight loss study, make it simple. You'd measure their weight at baseline, their weight at 4 weeks, weight at 8 weeks, weight at 12 weeks. You would take the baseline weight minus the 12-week weight and compute the difference, and that would be your outcome. In a longitudinal analysis, you would fit a model, for instance, with a mixed model or some other type of longitudinal modeling method that would, in essence, kind of measure the slope as you go from baseline to 4-week to 8-week to 12 weeks. So you're accounting for the 4 and the 8-week measurements as well as the 12-week measurement to model a line, and then estimating the change between baseline and 12 months from that line. It's not going to change your point estimate as much, but it will definitely make a difference in your variance because instead of just using the baseline and the 12-month measurement, you're now modeling it in such a way that you're accounting for the 4 and the 8-week as well, so that you're getting more information in terms of the complete longitudinal nature of that individual. That could be particularly critical if it doesn't increase in a linear fashion, right? If it's quadratic, so maybe it goes up quickly and then flattens out, or it goes up slowly and then jumps up. You can take into account of that. So it's not just what was the change from baseline to 12 weeks, but how did the curves differ over time? So those are all the questions I see right now. Are there any additional questions?
I don't see any additional questions. If there are any after the webinar, please feel free to email us at ninds-ctmc-info@umich.edu, and we'll be able to respond. I see one other one that just popped up.
That was just a thank you.
Oh, okay. So thank you to everyone. Thank you, Dr. Coffey, again. Please consider going to our website to evaluate the webinar, and if you want CMEs, as stated before, please complete the evaluation. Our next webinar will be May 6th at noon Eastern Standard Time, and the topic will be creating specific aims page for an early phase clinical trial. So thank you, and hope you can join us next time.
Great. Thanks, everyone.